Hello everyone, today we are going to do the past year NEET questions for the chapter Sexual Reproduction in Flowering Plants, and today in this video let's complete the part 6. So let's begin with the first question, which was asked in the year 2000. Double fertilization leading to initiation of endosperm and angiosperms required. So before we go into the solution, what do you mean by double fertilization? It is a complex fertilization mechanism of flowering plants, and this process involves the joining of the female gametophyte with two gametes. That is why it is called as double fertilization. Okay, so again I'm telling you, it is a fertilization that is a characteristic of seed plants in which one sperm nucleus will fuse with the egg nucleus to form an embryo, another fuses with the polar nuclei to form endosperm. So you have two gamete finally. So it's a fusion of, therefore the right answer to this question is option B, fusion of two polar nuclei and a second male gamete. Okay, the second male gamete entering the ovary fuses with the two haploid polar nuclei to form a triploid primary endosperm nucleus, and this develops into a, what does it develop into? It develops into an endosperm. Okay, therefore the right answer to this question is option B. Now let's move to the next question here. In angiosperms, triple fusion is required for the formation of TASH. So in angiosperms, angiosperms are the ones which produce flowers. In angiosperms, one male gamete fuses with the egg, resulting in the formation of diploid zygote. The other male gamete fuses with the two polar nuclei, forming the, what does it form? It forms primary nuclei, primarily endosperm nucleus. This type of fertilization is called as double fertilization. So already this is something that we discussed in the earlier question as well. So in angiosperms, triple fusion, okay, is required for the formation of endosperm, which is option B. Next question here. Double fertilization is a characteristic of TASH, and this question was asked in the year 1993. I think all of you by now would have guessed the answer, because already we saw two questions about it. Yes, double fertilization is a characteristic of angiosperms, in which one male gamete will fuse with the egg and the other male gamete will fuse with two polar nuclei to form an endosperm. Next question here. Double fertilization and triple fusion were discovered by, okay, so we have four options here: Strasburger, Leeuwenhoek. So Leeuwenhoek, Anton von Leeuwenhoek, that is option C. If you see, he is associated with microbiology. The right answer to this question is option B, Navaschin and Guignard. Okay, in angiosperms, one male gamete fuses with the egg to form a diploid zygote. The process is called as syngamy. The other male gamete fuses with two molar nuclei to form the endosperm. The process is called as triple fusion. These two acts together, known as double fertilization, is a process that was first demonstrated by Navaschin and Guignard, which is option B, the right answer. Next question here. Double fertilization is a fusion of TASH. Very easy question, and this question was asked in the year 1991. So it is one male gamete with the egg and the other will be paired with the two polar nuclei, that is the secondary nucleus. Therefore the right answer will be option D. So continuously, if you see in this particular part, we are going on seeing about double fertilization, which is associated with angiosperms. Angiosperms are flowering plants and they are also said to produce seeds, while gymnosperms, they too produce seeds. So here if you see in angiosperms, the one male gamete will fuse with the egg to form a diploid zygote, and the other process, what happens in the second part, if you see, the other male gamete will fuse with the polar nuclei, two polar nuclei, and it forms a triploid primary endosperm. But the main action here is the formation of the endosperm. Okay, next question here. Micropyle of seed is involved in the passage of TASH, and this question was asked in the year 1988. So micropyle, I think we already discussed in the earlier part of the same chapter. So what is this micropyle? Can anybody just recall and see? I'm talking about microfile. See, a microfile in terms of botany, you call this as a small opening. Very simple. What is it called as? It is called as a small opening in the surface of an ovule through which the pollen tube penetrates, often visible as a small pore in the ripe seed. So this microfile of seed, if you see, it is involved in the passage of water. It will help in absorbing of water during germination. Therefore, the right answer is option C. So here you have the micropyle. Okay? Yeah. Next question here. Functional megaspore in an angiosperm develops into dash. So here if you see, the first cell of the female gametophytic generation in angiosperm is a megaspore. It undergoes 3 mitotic division to form 8 nucleated and 7-celled embryo sac. Therefore, the right answer to this question is option B, embryo sac. So what are the other points that you can relate here when we talk about embryo sac? See, when we talk in terms of botany, an embryo sac is an oval structure within the ovule of the angiosperms which will contain the egg. Together with the fertilized egg, it will develop into a seed. The female gametophyte of the seed plant, consisting of thin walled sac within the nucleus that contains the egg nucleus and the other nuclei, which gives rise to endosperm on fertilization. So the functional megaspore in an angiosperm will develop into an embryo sac, which is option B. Next question here. Non-albinous seed is produced in dash, and this question was asked in the year 2014. Now, first of all, what do you mean by non-albuminous seed? We need to discuss that first. Now, when you take albuminous seeds, they are seeds which have food stored in a special nourishing tissue which is called as endosperms that remains persistent till maturity. Now, the seeds which store food materials in their cotyledons are when we talk about non-albuminous seeds, they are seeds which have the stored food on the cotyledons in a special structure which is called as kernel. Okay, I'm just giving you the difference between both. So albuminous seeds, they have this food stored in a special nourishing tissue which is called as endosperm. Okay, and when we say non-albuminous seeds, they have the food stored in a special structure which is called as kernel. Okay, yeah, it's called as kernel. So albuminous seeds refers to the seeds that retain or restore some part of the endosperms, while non-albuminous seeds refers to those seeds that consume their entire endosperm during the embryonic development. And you can take some examples of non-albuminous seeds, peas, groundnut, and all that. Therefore, the right answer to this question is option D, P. Okay. Next question here. Perisperm differs from endosperm in dash, and this question was asked in the year 2013. Perisperm is the remnants of the nucleus which is diploid. So when I say diploid, you always have to remember it is 2n, right? But endosperm is triploid. When I say triploid, it is 3n. Perisperm occurs in the seeds of black pepper, coffee, castor, cardamom, etc. Endosperm is a food-laden tissue which is meant for nourishing the embryo of the seed plants. While if you take this condition in angiosperms, the endosperm is formed as a result of what kind of fertilization you see there? You see there vegetative fertilization. Okay, triple fusion or fusion of the male gamete with the diploid secondary nucleus of the central cell. Therefore, perisperm differs from endosperm being diploid tissue, which is option B. Next question here. Seed coat is not thin, membranous in dash. Very easy question. Very easy question. It's a direct question. Just logical thinking would do. The seed coat develops from the integuments originally surrounding the ovule. It is thick and hard and coconut which protect the embryo from the mechanical injury and from drying out. Therefore, the seed coat is not thin and membranous in coconut. Next question here. The viability of the seeds is tested by dash. So this I think many might not be aware of. I'll give you the answer. The right answer is option C, 2,3,5-triphenyl tetrazolium chloride. So we have dehydrogenase enzymes present in the living tissues reduce the tetrazolium chloride to form as in a reddish water insoluble compound. So what happens here? These reactions occur in or near living cells which are releasing hydrogen in their respiration process. Viable tissues produce normal red color, weak living tissues produce an abnormal color. Dead tissues do not stain, remaining usually, so how do they remain? They remain usually white. So how can you test the viability of the seeds? You test it with the help of 2,3,5-triphenyl tetrazolium chloride. So the mechanism here it involves is, if it is a viable tissue, it will produce red color. If it is a weak one, it will remain without any stained process. It remains white, they say. Therefore, the right answer is option C. Next question here. Albuminous seeds store their reserve food mainly in dash. I think this is something that we already discussed. We saw non-albuminous seeds where they store their food in a special structure which is called as kernel. And here albuminous seeds store their reserve food mainly in endosperm, which is option B. It's the nourishment that it gives for the seed while germination. So it's a nutritive tissue which provides nourishment to the embryo in the seed plant. They retain the part of the endosperm, while if you take non-albuminous seeds, the whole endosperm will be consumed before germination. Okay, therefore the right answer is option B. Next question here. A drupe develops in dash. Again, this is an easy question. So when we talk about drupe, some fleshy fruits such as mango, plum, etc. usually have single hard stone that encloses a seed, and that is called as drupe. So I've given you the clue now. Yes, the right answer is option A, mango. Okay, so with this we complete sexual flowering in plants, and this is part 6. Hope to meet you in the next part with another set of questions. Thank you.